Hello everyone and welcome to the new session of the biology lecture series, and today we'll be solving the NCERT Exemplar questions for the topic sexual reproduction in flowering plants. I would like to mention that all the questions in this session are from the NCERT book. They are completely NCERT based. So today we'll be solving the very short answer type questions. So let's start with the first question. The first question is, you have to name the component cell of the egg apparatus in an embryo sac. So embryo sac consists of an egg apparatus, alright, which consists of egg cell and the synergid cell. And these egg apparatus is present at the micropylar end. And then at the chalazal end of the embryo sac, antipodals are present. And in the center, what are present? The two polar nuclei. Okay. So here, you can see the image of an embryo sac. So this is an embryo sac. Alright, here you can see at the chalazal end, 3 antipodal cells are present. Okay, in the center, polar nuclei are present. So these antipodals are haploid. Polar nuclei are also haploid. There are 2 polar nuclei here, you can see. Okay, and then at the micropylar end, we have the egg apparatus. Now, egg apparatus is made up of, you can see, an egg cell, which is again haploid. Then we have synergids. So 2 synergids are present. And both these synergids are haploid. And then within the synergids, filiform apparatus are present. Alright. So what was the question? What are the components of the egg apparatus? Okay. So the components of the egg apparatus in the embryo sac include 2 synergids, 1 egg cell, and the filiform apparatus. Okay, what is filiform apparatus? Filiform apparatus is nothing but it is a thickened structure, thickened cellular structure at the micropylar end. Okay, thickened cellular structure which is present at the micropylar end. Okay. And what is the role of filiform apparatus? It helps the pollen tube to reach the embryo sac. So pollen tube, okay, or you can simply say the pollen tube enters the embryo sac okay, from the micropylar end. Okay, you have to remember this pollen tube enters from the micropylar end. First, it enters one of the synergid that contains the filiform apparatus. Okay, so pollen tube enters the embryo sac through the filiform apparatus. Okay, that is present within the, where is filiform apparatus present? That is present at one end of the synergid, or you can say within the synergid. So filiform apparatus is attached to one side of the synergid. Here it is present, filiform apparatus. Okay, so these were the component of the egg apparatus. Alright, so let's start with the second question now. So second question is, you have to name the part of the gynoecium that determines the compatible nature of the pollen grains. Okay, so gynoecium, you already know, it is known as the female reproductive part. Okay, it is the female reproductive part in plants. That is known as gynoecium. So gynoecium can be represented by the pistil. Okay, pistil, or you can also call it as carpel. Now, we have already discussed about the structure of the pistil. So it consists of the ovary, style, and stigma. So I'm drawing a very rough diagram of the gynoecium. So it looks like this. At the base, it has a swollen ovary. Okay, so the basal part is known as the ovary and it is swollen. And then there is a tube-like structure which is known as the style, and the upper part is known as the stigma. So this region is the ovary, it is swollen, and ovary consists of the ovules. So ovules are present within the ovary. Alright, this part, this is known as the style. And the uppermost part, this is known as the stigma. Stigma is the receptive part. Okay, why it is called receptive part? Because it receives the pollen grain. And here, the compatibility or the incompatibility of the pollen grain depends here. Alright, so this is the receptive part and it receives the pollen. Okay, now coming back to the question again, you have to name the part of the gynoecium. Alright, parts of the gynoecium are stigma, style, and the ovary. Okay, and that part should determine the compatible nature of the pollen grain. Now let's have a look at the answer here. So this is a thinking process that I have given. This will help you understand this question and it will help you answer this question as well. So stigma, I've already told you, it is the part of the pistil. So what happens? So somewhere it is also mentioned as the part of the gynoecium. So pistil and gynoecium, you can use it as synonymously. You can say pistil represents gynoecium or carpel represents gynoecium. So somewhere it is also mentioned as the part of the gynoecium that determines the compatible nature of the pollen grain. Okay, so you can say stigma determines the compatible nature of the pollen grain and stigma it is one of the part of the gynoecium. Now, the pistil has the ability to recognize the pollen whether it is of the right type, means is it compatible, or it is of the wrong type, means it is incompatible. Alright, so the stigma, which is the part of the pistil, it determines whether the pollen is compatible or it is non-compatible. Now, if it is of the right type, means if it is compatible, the pistil accepts the pollen and promotes the post-pollination events that lead to the fertilization. Alright, so if the pollen is compatible, if it is of the right type, then pollen will germinate and it will produce the pollen tube, and the pollen tube will carry the two male gametes, which will then fertilize the egg cell. Alright, so what happens here? When the pollen lands on the stigma, if this is the stigma here, then the pollen will germinate and it will form the pollen tube. So this is the pollen tube which travels through the style. Okay, so this is the style and then it finally reaches the ovary. Okay, so this is the ovary, this is the style, and this is the stigma. This is the pollen. Okay, and it forms the pollen tube. So this is the pollen tube, which will carry the two male gametes, which will fertilize the egg cell present in the embryo sac. Alright, now if the pollen is of the wrong type, okay, if the pollen is incompatible, the pistil will reject the pollen. Alright, clear? Now, the ability of the pistil to recognize the pollen is followed by its acceptance or its rejection. So, it is the result of continuous dialogue between the pollen grain and the pistil-mediated chemical components of the pollen that interacts with those of the pistil. Okay. So, what happens when the pollen lands on the stigma? Then, what happens? there are some chemicals released by the pollen grains. And these chemicals are, or the synthesis of, or the generation of these chemicals, this is stimulated by the pistil. And these chemical compounds are then recognized by the stigma, and then the incompatibility or the compatibility of the pollen is decided. Okay, this is clear. Now let's talk about the next question. Here you have to name the common functions that the cotyledon and the nucellus perform. So we have already discussed nucellus is the part of the embryo sac, and cotyledon, it is a part of the seed. So cotyledons, these are present in the seeds. If you talk about the dicots, it has 2 cotyledon, monocots have 1 cotyledon. And nucellus, so nucellus, it is a part of the embryo sac. So we have already discussed about the structure of the embryo sac. It consists of the antipodal cell, it consists of the egg apparatus, which consists of the egg cell and the synergids. And in the center, the polar nuclei is present. Okay, and the embryo sac, you can say, it is present in the nucellus. Now, what are the common functions of this cotyledon and nucellus? Okay, so the common function that cotyledons and nucellus perform are as follows. So first function is the storage of the reserved food material. So both cotyledons and the nucellus, they store the reserved food material that can be used up later for the growth. Okay, so reserved food material stored in the cotyledon can be used for the growth of the seeds, whereas the reserved food material stored in the nucellus can be used for the growth of the embryo sac. Okay, next, nourishment. It also provides nourishment. Why? Because it stores the food material. Now these cotyledon, they nourish the growing embryo which is present in the seed, and the nucellus, it nourishes the embryo sac. Okay, so embryo sac is like this. This is an embryo sac. This is the chalazal end. This is the micropylar end. At the micropylar end, what are present? 2 synergids and 1 egg cell. So these 2 are synergids. Okay, and the upper one is this one is the egg cell. In the center, 2 polar nuclei are present, which fuse together later on, and then here 3 antipodal cells are formed. Okay, these are antipodal cells. Alright, so this is the nucellus. Alright, this is the nucellus, which gives nourishment to the embryo sac and it stores the reserved food material. Now moving on to the next question. Here you have to complete this flow chart. So here pollen mother cells, okay, which will be diploid. So with the help of meiosis, it produces pollen tetrad. Then after the formation of pollen tetrad, pollen grains are produced, and the pollen grain, they further divide to form the vegetative cell, and you have to answer the other one, which kind of cell is formed. Okay, let's have a look at the answer here. So pollen mother cells, they form the pollen tetrad and then they form the pollen grains. Let's see how these pollen grains are formed. Alright, and the pollen grain later divides into one vegetative cell and the other one is the generative cell. So generative cell is the answer to this question here. The pollen grain later on divides into a large vegetative cell and a smaller generative cell. Okay. Now what happens? The process of formation of the microspores from a pollen mother cells through meiosis is known as microsporogenesis. Okay, so pollen mother cells, these are diploid. Means they consist of 2 sets of chromosomes. So these are diploid. So we can write here, these are pollen mother cells. These are diploid. And with the help of the process known as meiosis, So meiosis is known as reductional division. It will reduce the number of chromosomes to half. So after meiosis, what are formed? Microspore. So microspore tetrad is formed. Okay, because these microspores, they are arranged in a cluster of 4 known as microspore tetrad. Okay, so you can write here a microspore tetrad, or you can write pollen tetrad, because microspore later forms pollen. So you can also call it as pollen tetrad, and the cells here will be haploid, means N. Then what happens after this? The pollen tetrad, here what will happen? The microspores present in this tetrad will then form the pollen grains. And the pollen grain will then divide to form one vegetative cell and one generative cell. Okay. So if this is the pollen grain, this large one will be this is the larger cell. This is the vegetative cell. The smaller one is the generative cell. Now, as the anther matures and they dehydrate, the microspores, they dissociated from each other and they develop into the pollen grain. So microspores which are present in the tetrad, then they, as the anther matures, they dehydrate and they disperse and they dissociate from each other and they form pollen grains. And pollen grains are also known as the male gametophyte. Now, when the pollens mature, it contains 2 cells. One is the vegetative cell, another one is the generative cell. Okay, so vegetative cell is bigger and generative cell is smaller. Okay, this is clear. So this is an image showing you the first a microspore tetrad. Here you can see in the image A. Here you can see 4 microspores. This is 1, 2, 3 and 4. These are 4 microspores that are arranged in a tetrad. That's why we call it as a pollen or a microspore tetrad. And then what happens as the anther matures is dehisces and each microspore then forms the pollen grain. So here in the image B, you can see a microspore maturing into a pollen grain. Here you can see this is the pollen grain. It contains the nucleus. Then in the second stage, there is a development of vacuoles also. Okay. In third stage, you can see there is a division. So here you can see formation of vegetative cell and the generative cell. And in the mature pollen, so we can say this is the mature pollen. In this mature pollen, the larger cell, so this one is known as the vegetative cell and the smaller one is known as generative cell. So this is a pollen at 2-celled stage. Okay, you can see 2 cells are formed. So this is the pollen at 2-celled stage, and most of the pollens are released at this stage, 2-cell stage. Sometimes some of the pollens, or you can say in some of the plant species, the pollens are released at 3-cell stage, where the generative cell will further divide and it will form the 2 male gametes. That is known as the 3-cell stage. Alright, this is clear. Okay, let's start with question number 5. Here you have to indicate the stages where meiosis and mitosis occur. So in the following flow chart, they have given you the following stages. First is stage number 1, stage number 2 and stage number 3. Okay, so megaspore mother cell, you know it is diploid. Okay, and megaspores are haploid. So which kind of division should take place? Here meiosis should take place. Okay, meiosis is also known as the reductional division. So megaspore mother cell undergo meiosis to form megaspores, and initially 4 megaspores are formed. Okay, 4 megaspores are formed which are haploid, and out of these 3 degenerate. 3 degenerate and only 1 megaspore, okay, it undergoes mitotic division. So second stage will be mitosis. Okay, here mitotic division will take place and it will form the embryo sac. Then embryo sac again will undergo mitotic division to form the component of the embryo sac, such as egg cell and all, and antipotals and synergids. Okay. So what should be the answer? Megaspore mother cell undergo meiosis to form megaspore, which are haploid, and then megaspore undergo mitosis to form the embryo sac, and then here it should be mitosis, undergo mitosis to form the egg cell, which is haploid, because meiosis can only take place in the diploid cells. Okay, mitosis takes place in the haploid cell. So here it should be mitosis. Okay, now let's understand about this flowchart. The diagram, the diploid megaspore mother cell, which are also known as MMC, undergo meiosis to form a linear tetrad of 4 haploid megaspores. This I've already told you. Now, 3 mitotic divisions inside the functional megaspore. So 3 megaspores degenerate. So out of the 4, 3 megaspores degenerate and only 1 function megaspore is left, which undergo mitosis to form the embryo sac. Okay, in which 8 haploid nuclei are present, while the other 3 megaspores degenerate. Alright, so embryo sac, it is an 8 nucleate structure. Okay, this you already know. Embryo sac in the angiosperms is an 8 nucleate structure and 7 celled structure. Alright, so this is clear. Now, the embryo sac is a 7 celled and a 8 nucleated structure. Okay, 3 micropylar, 3 chalazal and 1 central. Okay, now the 3 micropylar cells are collectively known as the egg apparatus. So egg apparatus consists of egg and these 2 synergids, so means it consists of 3 nuclei. Okay, it consists of 2 synergids and 1 egg cell, whereas the cells at the chalazal end. Okay, so cells at the chalazal end are known as the antipodals. These antipodals are also haploid and they are 3 in number. Okay, and in the center, what is present? So in the center is present again 2 polar nuclei. Okay, so 3 plus 3 is 6 plus 2 means 8 nuclei. 8 nuclei are present, but what happens later? These 2 polar nuclei, they fuse with each other. Okay, that's why we call it 8 nuclei and 7 celled. Alright, this is clear. Now, the 3 chalazal cells are known as or form the antipodal cell. The central cell is in the form of 2 nucleated cell. Okay, till the fertilization occur and is called as polar nuclei. This is clear. So here you can see a diagrammatic representation of the mature embryo sac. Okay, at the chalazal end, antipodals are present. Okay, so 3 nuclei from here. Polar nuclei. Here we have 2 polar nuclei. Okay, and these 2 fuse later to form the central cell. At the micropylar end, egg apparatus is present, which consists of 2 synergids and which consists of 2 synergids and 1 egg cell. Okay, so here 2 synergids are present. So in total, egg apparatus consists of 3 nuclei. Okay, so 3 plus 2 plus 3 is 8 nuclei. Okay, now let's talk about question number 6. In the diagram given below, show the path of the pollen tube. Okay, so pollen germinates to form the pollen tube. Okay, from the pollen tubes are formed from the pollens. Okay, so in this diagram, this question is asking that in the diagram given below, show the path of the pollen tube from the pollen on the stigma into the embryo sac and name the components of the egg apparatus. Okay, so this is what is this? This is a diagrammatic representation of a pistil. Here you can see the swollen part is known as the ovary. Ovary consists of what? It consists of the ovule. Okay, this is known as the style through which the pollen tube passes, through which it is the passage for the pollen tube. And here this part is known as the stigma. And here you can see the pollen lands here. So suppose this is the pollen. This pollen, if compatible with, will germinate and it will form the pollen tube. Okay, and the pollen tube will start traveling from here and it will carry the two male gametes. Okay, the pollen tube will it will enter the style and then it will enter the ovule. Okay, in the end, it will enter the ovule through which part? From the micropylar end. Okay, so the pollen tube enters the ovule through the micropylar end. Okay, now following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube. So first pollen tube is formed. Okay, now the contents of the pollen tube, which are the two male gametes. Okay, they move into the pollen tube. The pollen tube grows through the tissue of the stigma, okay, and reaches the ovary. Okay, through which? Through style. Now, after reaching the ovary, the pollen tube enters the ovule through the micropylar end. Okay, and then enters the embryo sac. Okay, but when the pollen tube enters through the micropylar end, what happens? First, it reaches the micropylar end and then it enters the embryo sac through what? Through the synergids. Okay, so first pollen tube will reach the micropylar end and through the synergid. Okay, and within synergid, we have already discussed, are present the filiform apparatus. Okay, the pollen tube will reach the micropylar end. It will enter the synergid. It will interact with this filiform apparatus. Filiform apparatus are present within the synergids, and then it can release the male gametes. Okay, so when the pollen tube enters the synergid through the filiform apparatus, then what happens? It leads to the degeneration of the synergid. Okay, this is clear. Now the pollen tube breaks to release its content, which are the two male gametes. Out of the two male gamete, one fuses with the egg cell, this you already know, and the other fuses with the central cell. Okay, so this, you know, this kind of fertilization is known as double fertilization. This is known as double fertilization. One male gamete fuses with the egg cell and the other male gamete fuses with the central cell. Okay, so when the egg cell fuses with the central cell, it is known as triple fusion. So you can write, when the male gamete, it fuses with the central cell, okay, which is already diploid. This phenomena is known as the triple fusion. And you can see here, fertilization is taking twice, that's why it is known as double fertilization. So this happens in angiosperms. Here again you can see diagram of a pistil. Alright, you can see pollen land on the stigma. Okay, and if they are compatible, they form the pollen tube. So this is the pollen tube. Okay, now this pollen tube travels through the style and you can see the path here, then it enters the ovule through micropyle. Okay, so this is the micropylar region where synergid cells are present. So this is the micropylar region. Okay, the pollen tube will enter the ovule through the micropyle. First, it will enter the filiform apparatus, which is present in the synergid cell, and then it will release the two male gametes. Okay, two male nuclei are present here. You can see in the pollen tube. Then, when the pollen tube enters the ovule, it will break and it will release the two male nuclei. One will fuse with the egg cell and other will fuse with the central cell, which is formed by the fusion of the two polar nuclei. Okay, here you can see the polar nuclei. Here you can see the antipodals. This is the pollen tube. Okay, so this is the path of the pollen tube through the pistil. Okay, so this diagram shows you the longitudinal section of a pistil of a flower. Now, if you talk about the components of the egg apparatus, this also we have already discussed. It consists of two synergid, one egg cell, and one filiform apparatus. Okay, again, this is the image showing you the embryo sac. Here you can see the micropylar end. Egg apparatus consists of what? Two synergid cell, and here you can see filiform apparatus within the synergid and pollen tube. First, enter the filiform apparatus which is present within the synergid. So egg apparatus is made up of synergid, filiform apparatus, and the egg cell. Okay, so these are the components of the egg apparatus. Okay, now let's move on to the next question. Now here you have to name the parts of the pistil which develop into fruit and a seed. Okay, parts of a pistil. Pistil you already know. It consists of stigma, style, ovary, and within ovary what is present? Ovule. Now you already know. Okay, ovary develops into the fruit. So ovary after fertilization develop into fruits, and ovule after fertilization develop into seeds. Okay. Now, pistil is the female reproductive organ. This we already know, that receives pollen to fertilize the egg located in the ovary. A pistil has three parts: stigma, which receives the pollen, which grows down through the style. So pollen tube grows down to the style and it reaches the ovary. Now ovary contains the ovule, which contain an egg. Okay. Now the ovary develops into the fruit after fertilization, and ovule develop into the seeds after fertilization. Okay, so this was the answer to this question. Here you can see stigma, style, ovules are present within the ovary. Okay, this is thalamus, which is the swollen part of the flower. Okay, so this is the part of the flower. Ovary develops into fruits and ovule develops into seeds. Okay, these are the parts of the pistil. Now let's talk about question number 8. Now, in case of polyembryony, if an embryo develops from the synergid and another from the nucellus, which is haploid and which is diploid. So which one will be haploid and which one will be diploid? This you have to answer. Okay. Now first you have to know what is polyembryony. So the presence of more than one embryo in a seed is known as polyembryony. Example of some plant where polyembryony is seen is lemon, groundnut, okay, and some other plants. This is known as polyembryony. Okay. If the embryo developed from the synergid, okay, so synergid you know is a haploid cell. So embryo developed from synergid, okay, through mitosis will be haploid, as the ploidy of the synergid is haploid, okay, and embryo developed from the nucellus is diploid, as the ploidy of the nucellus is diploid. Okay, this is very simple. Nucellus is diploid. Its ploidy level is 2n. It consists of 2 chromosome sets, whereas synergid consists of 1 chromosome set. It is haploid. So from nucellus, embryo which will develop will be diploid, and from synergid, if the embryo develops through mitotic division, it will be haploid. Now let's talk about question number 9. Can an unfertilized apomictic embryo, they are talking about apomictic embryo sac, give rise to diploid embryo? If yes, then how? You have to explain. Okay. Yes, if megaspore develops into the embryo sac without meiotic division, okay, then the egg will be diploid. Obviously, because meiosis, what it do? It reduce the number of chromosomes. Okay. So the diploid egg develops into an embryo later by mitotic divisions. Okay, so this is clear. Now, apomixis, if we talk about apomixis, it is the formation of seed without the embryo. So it is different from the parthenocarpy. Apomixis is the, if you define apomixis, it is the formation of seeds without fertilization. Okay, there is no fertilization here. Okay, that's why it is a form of asexual reproduction in which seeds are produced without the fertilization. Now, question number 10. Which are the 3 cells found in a pollen grain and when it's shed at the 3-cell stage? So this I've already told you. Pollens, they are shed at the 2-cell stage or they are shed at the 3-cell stage. Okay. Now let's have a look at the answer. Now in over 60% of the angiosperms, pollen grain are shed at the 2-cell stage. What are the 2 cells present within the pollen? First is the vegetative cell and second is the generative cell. This also we have already discussed. Now in the remaining species, the generative cell divide mitotically to give rise to 2 male gametes. Okay, before pollen grains are shed. Okay, at the 3-cell stage. So now the cell, what are the 3 cells present at the 3-cell stage? First is the vegetative cell. Then we have the 2 male gametes. Okay, so if we talk about 2-cell stage, pollen can be released at 2-cell stage. So in majority of angiosperms, in over 60% of angiosperms, the pollen grains are shed at 2-cell stage and they consist of vegetative cell and another one is the generative cell. If they are released at the 3-cell stage or they are shed at the 3-cell stage, what are the 3 cells here? One is the vegetative cell and the generative cell. It divides to form 2 male gametes. Okay, so these are the 3 cells. All right. Now, question number 11. What is self-incompatibility? Okay, now let's have a look at the answer. It is a genetic mechanism which prevents the self-pollen from fertilizing the ovules. Okay, so self-incompatibility, it simply you can write it prevents self-pollination. Okay, it prevents the self-pollination. And what does it do? It promotes cross-pollination. Okay, so prevents the self-pollen means prevents the self-pollination. What is self-pollination? When the pollen from the anther, okay, is transferred to the stigma of the same flower present on the same plant. Okay, this is known as self-pollination. Okay, now there is a prevention of the self-pollination, okay, from fertilizing the ovules. The pollens are not able to fertilize the ovules. And how this happens? It happens by inhibiting the pollen germination. So either the pollen doesn't germinate or the pollen tube growth in the pistil is inhibited. Okay, so either the growth of the pollen is inhibited or the growth of the pollen tube is inhibited. Okay, and that's how self-fertilization, self-pollination is prevented. Now let's talk about question number 12. Now name the type of pollination in self-incompatible plants. Self-incompatibility we have already discussed. Okay. Now let's have a look at the answer. In self-incompatible plants, okay, while self-pollination is incompatible, means it prevents the self-pollination. Cross-pollination will occur, obviously. Okay, so this I've already told you. In self-incompatible plant, as the self-pollination is prevented, and you have seen either the pollen tube doesn't germinate or it forms, or there is an inhibition of the pollen germination. So because of this, cross-pollination is promoted. Okay. Now, self-incompatibility is a genetic mechanism, this we have already seen, which prevents the self-pollen from fertilizing the ovules by inhibiting either the pollen germination or inhibiting the pollen tube growth in the pistil. Now let's talk about question number 13. Now here you have to draw the diagram of a mature embryo sac and show that it is 8 nucleated and 7 celled in nature. Okay. Now show the following parts. While you draw the diagram of the mature embryo sac, you have to show this following part. You have to show the antipodals, you have to show the synergid, egg cell, central cell, and the polar nuclei. So here they're talking about the mature embryo sac. Okay, this you have to remember. So this is easy. We have already discussed this so many times. So this is the image. So here you can see this is an embryo sac. This is a typical embryo sac of a angiospermic plant. So at the chalazal end, you can label here. At the chalazal end, what is present? Antipodals are present, which are 3-celled with 3 nucleus. Okay. Then at the micropylar end, so this is the micropylar end. This you have to remember. Which is micropylar end, which is chalazal end. Here egg apparatus is present. An egg apparatus we have seen consists of 2 synergid and 1 egg cell. So it is also 3-celled with 3 nucleus. Okay. Now let's talk about the polar nuclei and the central cell. So here in mature embryo sac, what happens? These polar nuclei, they will fuse. They fuse with each other. Okay. That's why it is 1-celled and 2 nucleated structure. So because of this, what happens? The embryo sac, what is the nature of the embryo sac? It is 8 nucleated. It is 8 nucleated and it is 7-celled. Okay, this is clear. So the mature embryo sac is 7-celled and 8 nucleated structure. Okay, this is clear. Now let's talk about question number 14, which is the triploid tissue. Means the ploidy level here is 3N. Means it contains 3 chromosomal set. Okay, 3 chromosome set. So which is the triploid tissue in the fertilized ovule? So they are talking about fertilized ovule. In fertilized ovule only this triploid tissue will be present. And how this triploid condition is achieved? So we have already discussed about the triple fusion. What was triple fusion? When one of the male gamete fuses with the central cell. Okay, then triple fusion or a triple tissue is formed, or a triploid tissue is formed. So one of the male nucleus, male gamete, it fuses with the central cell, which is diploid. Why it is diploid? Because it's formed by the fusion of the 2 polar nuclei. So as a result, a triploid tissue will be formed. Let's see. Let's have a look at the answer. So this triploid tissue is known as endosperm. Okay, so endosperm is the triploid tissue. Okay, it results from triple fusion involving union of one male gamete with the two haploid polar nuclei. So these 2 polar nuclei fuse to form the central cell. Okay, so this central cell fuses with the one of the male gametes and forms a triploid endosperm. So in this figure, you can see after fertilization, what happens? Okay, synergists, they degenerate. Okay, zygote is formed. How zygote is formed? When one male, so initially 2 male gametes are released. So one of the male gamete, it will fuse with the egg cell, and then it will form the zygote, which is diploid. Okay, and the other male gamete fuses with the central cell. Okay, fuses with the central cell, and initially it forms a primary endosperm nucleus, which is triploid, and this primary endosperm nucleus will later form the endosperm. So endosperm will be triploid, and after fertilization antipodals also degenerate. So this is an image showing you the fertilized embryo sac, showing the primary endosperm nucleus, which is triploid, and this will give rise to the triploid endosperm. Okay, so this is clear. Now let's move on to the next question. Are pollination and fertilization necessary in apomixis? Apomixis, you already know, is the formation of seeds without the fertilization. Okay, so no fertilization is happening, means pollination is also not required. So you have to give reasons. Okay, now first understand what is apomixis. The phenomena of asexual reproduction. So it is a kind of asexual reproduction that mimics the sexual reproduction. And how does it mimic sexual reproduction? By formation of seeds. So through sexual reproduction also seeds are formed. Here through apomixis also seeds are formed. But the difference is here in apomixis seeds are formed without the fertilization. Okay, so apomixis is a kind of asexual reproduction which mimics the sexual reproduction by formation of seeds. Okay, now this is the answer. Pollination and fertilization are not necessary for apomixis. Okay, the reason in support of this are given below. Now let's have a look at the reasons. First one, when embryo sac develops from the megaspore without the reductional division, means without the meiosis. There is no meiosis. The egg is diploid, obviously, then the egg will be diploid, and it develops into a diploid embryo. Okay. Now, second point, embryo sac can also develop from the diploid nucellus cells, in which case egg is diploid that develops into embryo parthenogenetically. Okay. So here pollination and fertilization are not required. But what is happening here? There is no meiosis happening. Okay. No reduction division is happening. Okay. And the megaspore in some cases can, okay, without undergoing meiotic division, can form the egg cell, which will be diploid, and then egg can further develop into a diploid embryo. And through this seeds will be formed. Now again, if embryo sac develops from the nucellus, so nucellus cells are always diploid. Okay, so these are diploid. So in some cases, nucellus can also form the embryo sac through mitotic divisions. So here also there is no meiosis. Okay, and then what happens in this case? Egg will be diploid, and then it can form the embryo parthenogenetically, and here also embryo will be diploid. Okay, so this happens during the apomixis. No fertilization and pollination are required. So this is clear. So let's start with question number 16. So question number 16 is, we have to identify the type of carpel with the help of diagrams given below. So these are the two diagrams that have been given. So this is the first diagram, this is the second diagram. Okay. Now the gynoecium, if we talk about the gynoecium, which is the female reproductive part, it represents the female reproductive part of the flower, and it consists of single pistil. Okay, when single pistil is present, then it is known as monocarpellary. Mono means one. Okay, so monocarpellary means one carpel. Or the gynoecium may be a combination of many pistils. Okay, so when more than one pistil is present or many pistils are present, then it is known as multicarpellary. So multi means many. So many carpels can comprise a gynoecium. Okay, Now, let's talk about the first image. So, if the gynosium has multiple carpels fused into a single structure, okay? So, first point is that if the gynosium consists of many carpels, but these carpels are fused together to form a single structure, it is known as syncarpous gynosium. Syn means fused, all the carpels are fused here. So in the given diagram, this type of carpel is syncarpous. An example of plant where syncarpous gynosium is observed is the poppy plant. Okay, here you can see this is the syncarpous ovary. The carpels are fused together. Syn means fused. Okay, so here is the ovary. This is the stigma here, which is the receptive part. It receives the pollen. Okay, this is multicarpellary syncarpous pistil of papaver. Okay, papaver somniferum. What is the common name of papaver somniferum? It is opium poppy. Or you can just simply say poppy. Now, let's talk about image B. So, if the gynosium has multiple carpels in the free form, it is apocarpous. So, apo means free. Okay, so here first condition is multicarpous carpels are present, but they are in free form, they are not fused. So, this is known as apocarpous condition. So, image B, this type of carpel is known as apocarpous gynosium. Now, example of plant where this apocarpous gynosium is seen is Michelia. Okay, here you can see carpels are there, multiple carpels are there, but these carpels are free, they are not fused. Okay, so this is the multicarpellary apocarpous gynosium of Michelia. Okay, so it is a scientific name. So scientific name should be italicized or it should be underlined. Okay, now let's talk about question number 17. How is pollination carried out in water plants? Water plants means aquatic plants that lives in water. Okay. Now first, in many aquatic plants with emergent flowers, pollination occurs by wind and insects. Okay. This you already know. In aquatic plants with emergent flowers. Okay. Where flowers are above the water, pollination occurs by wind or by insects. So wind is a abiotic factor. Okay, or you can say abiotic pollination. Insects, okay, it come under biotic pollination. All right. So here, although the plant is aquatic, means it lives in water, but the flowers are above the water. They are, okay, emergent flower. These are known as emergent flower. Now, in water plants, if pollination occur below the surface of the water, okay, so pollination can occur above the surface of the water or below the surface of the water. So if the pollination occur below the surface of the water, yani ki inside water, it is known as hypohydrophily. Okay, hypo means below. Hydrophily means pollination with the help of water. Hypo means beneath or under, or you can say beneath. Okay, for example, ceratophyllum. Okay, so in ceratophyllum, what happens? The flowers, they break and they reach the surface. What happens to ceratophyllum? Flowers, they break from the plant. Okay, the flower breaks and then they reach the surface. Reach the water surface. Okay, and then what happens? When they reach the water surface, what they happen? Then the pollens are released. Okay, then the anther dehisce. Okay, pollens are released. Okay, after entheogenesis, what will happen? Pollens will be released on the surface of the water. And then what happens? These pollens, they sink. They sink in the water and pollination takes place below the surface of the water. Okay, so this is known as hypohydrophily. Then let's talk about epihydrophily, means above the surface. So in water plants, if pollination takes place over the surface of the water or above the surface of the water, it is known as epihydrophily. Example of plant where epihydrophily is seen is Vallisneria spiralis. Now let's talk about the next question. Question number 18. What is the function of two male gametes produced by each pollen grain in the angiosperm? So you know pollen grain produce two male gametes. This you already know. And one male gamete fuses with the egg cell. Okay. So one male gamete, okay, fuses with the egg cell. And what it forms? It forms zygote. And then another male gamete fuses with the central nuclei, or you can also call it as secondary nuclei. Okay, so this is formed by the fusion of the two polar nuclei. In this case, what is formed? Endosperm, which is triploid. Okay, and this entire phenomenon is known as double fertilization. Okay, this is known as double fertilization, where one male gamete fuses with the egg cell, another female gamete fuses with the secondary nuclei, also known as the central cell. Okay, and here, from this fusion, from which endosperm is developed, this is known as triple fusion. This is known as the triple fusion or triple fertilization. Okay, now let's talk about this. A mature pollen grain contains two cells. First one is vegetative, second one is the generative cell. Okay, this we already know. Now, generative cell is the generation cell. Okay, that divides mitotically to give rise to two male gametes. This also you already know. Now, one male gamete unite with the egg cell, forming the zygote, which will form the embryo. Now, this is known as fertilization or the syngamy. Okay, now the other male gamete, it unites with the two polar nuclei, resulting in the formation of the endosperm. Okay, and this phenomena is known as triple fusion. Okay, this also we have already discussed. Okay, so this is clear. So these are the role of the male gamete. First, it helps in the formation of the embryo. Second, it helps in the formation of the endosperm. Okay, so this was the end of this session. We will discuss rest of the question in the next session. Thank you very much.